My name is Joe Lawdanski, and today I'm going to be telling you all about Anthracnose of Turf. Anthracnose of Turf has been causing turf cultural managers problems for years. It is caused by the pathogen Pathogenic Structures. It causes disease in 13, roughly, warm season and cool season grasses, mainly in the Agrostis species, which is bent grass and poa annua and other cool season grasses. Some symptoms seen in anthracnose: foliar lesions, and these foliar lesions tend to appear chlorotic at the basal root. And if you look closely at these foliar lesions, you can almost see the black acervili, but we'll get into that later. Some signs of this disease is these black setae, which are hair-like structures of the pathogen itself, and those are indicative of the fruiting body being present, which is the acervili. Those acervili contain the conidia, which are the asexual spores, which will then be released at some point in time and propagate. In terms of the disease cycle for it, it overwinters as mycelium or conidia on the leaf blades. The conidia serve as the primary inoculum. Mid-spring to early summer and during long wet periods, the conidia will germinate and a germ tube will form. The aprosorium will then form from that, and it will create a penetration peg into the epithelial cell, and you now have infection. The pathogen itself acts as a necrotroph, and really the best way to control it is good cultural practice. I mean, it requires 12 hours of continuous wet biofilm on the leaf in order to cause germination. So best way to do that is to water deep and infrequently in the mornings, and apply fungicide prior to the anthracnose even being present, or use a systemic fungicide in order to control it after infection. Now, as for my project, I decided to use clay models to represent the pathogenic structures. Now, each model that I made is almost a zoomed in look at the one prior to. So I started with the black acervili, which are the fruiting bodies, and those have the black setae on them as well, which are hair-like structures that are virulence factors for adherence. So here's my little acervili, and it has conidia within it at this point in time. Now when it comes time to release these asexual spores, this is our next model. This is what that would look like. That is the acervili opening up and your conidia spores being released from that opening. Next, we have a zoomed in look at our conidia. It is crescent shaped, and there are thousands of these at one time within those fruiting bodies, so it easily propagates, and it's mainly propagated by cultural practices such as mowing and foot traffic, things such as that. So, like I was saying, this has been causing problems for years for turf managers, superintendents, and best way to control it is just good turf practice, good irrigation practices, fungicidal applications, and fertilizer applications without over-fertilizing. Anyways, that is what I have for you today. Thank you for your time. My name is Joe Lawdanski. Have a good one.